by the University of Michigan. We are going to get started today. I'm Taylor West. I'm a second-year PhD student here at the University of Michigan in the Department of Ecology and Evolutionary Biology, and I will be moderating today's session. I wanted to make a couple quick notes before we start. First off, there is live transcripts available for the talks today. There is a button in your Zoom bar, you'll see a CC, and if you click on that, it will transcribe the videos for you. So we have that option for you. There's also a Q&A box in the Zoom bar, which we will be using. So if you have any questions for our speakers today, or questions about early career science and natural history collections in general, please put them in this box. And I also just want to note that the Q&A box is different from the chat box. So if you look below, there's chat and Q&A. Feel free to use both, but any questions that you would like the speakers to address, please put them in the Q&A. Okay, so today is our second Friday of 5 Fridays in a row that we're holding ECSS this year. And it's our first day with early career scientist speakers. So we are very excited to have with us Dr. Jocelyn Colella and Dr. Kelly Speer today. Jocelyn will be speaking first. And so I'm going to introduce her and she will give her talk, and then Kelly will give her talk next. So after I introduce Jocelyn, I also just want to point out that we will have a short Q&A session specifically for her talk right after she gives her talk. And then Kelly will give her talk and we will have a quick Q&A for her talk as well. And then after these are done, we will do an overall panel discussion with both of our speakers where we will cover larger, broader questions about collections and early career research. Okay, with that, I will introduce Jocelyn. Jocelyn Colella is a collections-based evolutionary biologist who uses mammals as models to understand how organisms change through time, with particular interest in conservation, adaptation, and hybridization. She received her PhD from the University of New Mexico in 2019, where her research focused on hybrid dynamics of high-latitude mesocarnivores. Jocelyn is now a postdoctoral research associate at the University of New Hampshire, where her current research aims to unite natural history collections with biomedical sciences by identifying genetic mechanisms of dehydration tolerance in desert-adapted rodents. And she does this to inform gene therapies in humans and the adaptive capacities of species to increasing desertification. So please help me welcome Jocelyn for her talk titled Connecting Next Generation Museum Collections to Public Health.
Great, thank you. Should I share?
Yep, you're good to go.
Can you all see that?
Looks great.
Perfect. Thank you so much for the introduction, and thank you to the rest of the committee for inviting me to speak at this symposium. It's pretty cool to be part of such a stellar lineup of early career scientists, and I'm flattered. As they mentioned, I am currently a postdoctoral research associate at the University of New Hampshire in the McManus Lab, but I will be starting a faculty curator position at the University of Kansas this coming August, and I'm pretty psyched. Today, I'm going to present on an issue that was timely last March, and ironically, or unfortunately, still is, and that is connecting next generation museum collections to public health. And I'll start with the most obvious connection. Zoonotic diseases. Zoonotic diseases are those that can move from non-human animals to humans. Presumably, you've all heard of coronavirus by now. If not, you can just look at the news. Hopefully some of you have been vaccinated, but we know coronavirus is a zoonotic disease. But other diseases like Lyme disease, Ebola, West Nile, and rabies are also zoonotic diseases. Mammals, which I study, are the most common host of zoonotic diseases due to our shared evolutionary history with them, as we too are mammals. And these diseases are of significant public health concern, as evidenced by the current and ongoing global pandemic, which has taken the lives of more than 100 million people worldwide and cost more than $20 trillion in this year alone. Museums are the biorepositories that hold the physical samples necessary to detect and monitor zoonotic diseases and their hosts through time. This includes estimating pathogen abundance, geographic distributions, infection rates, identifying reservoir and susceptible host populations, and many of the ecological and environmental factors that may contribute to future disease spillover events. Hantavirus provides a good example of the importance of museums in emerging infectious disease research and response. After a string of mysterious deaths in the American Southwest in the early 1990s, the CDC funded local natural history collections to survey small mammal populations to figure out what the disease was and where it was coming from. These surveys not only identified hantavirus as the agent responsible for recent deaths, but also identified deer mice of the genus Paramiscus as the reservoir host of the virus. But the best part of this example is that our understanding of the virus and its host didn't end after we knew what it was and where it came from. Thousands of specimens were archived as part of this project, and these specimens were used over and over again in primary research to discover additional mammalian hosts and viral strains, and that hantavirus outbreaks in humans are correlated with El Niño climate events. The El Niño Southern Oscillation leads to more rain in the Southwest. More rain leads to more food for small rodents like Paramiscus, and that leads to spikes in rodent population sizes and a high density of individuals. Just like in humans, a high density of individuals is the perfect storm for the spread of disease. And we can now use this information to anticipate the reemergence of hantavirus following El Niño climate cycles. This type of whole ecosystem observation is critical to mitigating future outbreaks. But it's also impossible without frequent sampling and resampling of mammal hosts through time, and open access to those samples through museums, of course. But unfortunately, this is still not the norm in many disciplines that use and collect wildlife samples. These types of integrative discoveries increasingly rely on next generation natural history collections. These collections prioritize the archival of extended specimens. I think Rob and Dan touched on this a bit last week, but extended specimens go beyond the immediate goals of a single project, say screening for the presence of a particular disease, coronavirus, for example. And instead, they aim to maximize the amount of information collected from a single animal or a single collecting or sampling event. This means that these types of collections collect traditional morphological materials like skins, skulls, and skeletons, but also detailed information on collection localities, ecology, and the environment. Frozen tissues for use in genetic and genomic research, like what I do. And all of these resources also contain viruses and pathogens that were present in that animal, as well as internal and external parasites and other lines of information. Traditionally, collected samples would be used for whatever they were originally collected for, right? And historically, these questions were mostly systematics or taxonomy based, which is great, but times are changing. And extended specimens, cumulatively collected by the scientific community and then archived and made available through museums, can be used not only for their original collection purpose, but also by other researchers in the community to answer new and different questions across disciplines, looking at things like physiology or phenology, all sorts of omics-based sequencing, community composition and interactions, pathogen distributions and associations, and how all of those variables change over time. Every field biologist doesn't have to be a genomicist and a virologist and a taxonomist and a whatever. Instead, next generation natural history collections connect samples to scientists to maximize the information gained from each sampling event. While these collections importantly allow us to quickly and effectively detect, identify, and combat emerging zoonoses, they also have many unintended uses. And this is where some of my research comes into the mix. and provides another example of an unintended and innovative connection between museums and public health. We all know that animals do extraordinary things. Just in mammals, camels can survive 7 months without drinking water. Polar bears can survive temperatures below -50 degrees Celsius. Weasels can wait years to reproduce after mating to ensure there are adequate resources for reproduction. And elephants are immune to cancer. In general, I think it's safe to say that humans would like to be able to do all of these things. So it's clear we have a lot to learn from the natural world. The materials and frozen tissues in museum archives are some of the best available resources for understanding how these animals accomplish these feats. As part of my postdoc research, I've used museum tissue samples to understand how mammals survive in hot deserts without water. As a conservationist at heart, I'm interested in this because it allows us to better predict organismal responses to global warming, where we anticipate more extreme bouts of intense heat and aridity, especially in areas like the American Southwest. But the National Institutes of Health, or NIH, is interested in this because humans have a very limited ability to survive desert conditions. 2.5 million people die of dehydration-related illnesses annually. Yet there are a number of mammals that are acutely adapted to desert life. Camels, of course, being the stereotypical example, but also rodents, lagomorphs, even carnivores, as well as non-mammals. Studying the genomes of these desert species may allow us to identify the molecular basis of complex traits, like dehydration or heat tolerance. Unfortunately, though, a number of these species are not yet well represented in natural history collections for one reason or another. Looking at the available sampling for each genus shown here, these are the total number of preserved specimens listed in the Global Biodiversity Information Facility, or GBIF. If I limit this search to the species level, it more than halves all of these numbers. Sufficient sampling is critical to ensuring that we have the statistical power to detect genetic outliers. But it's also important that samples are representative across space and time to account for population structure, mutation rates, and differences in selective regimes. Communities outside of museums are becoming more aware of museum sampling. And as these communities begin to contribute to the collective growth of museum collections, the available sampling base improves for the entire community. But until then, there are a ton of deer mice in biorepositories. Remember, this is the same genus that is a natural reservoir for hantavirus. And I think last week, Rob said something similar, noting not only how cool Peromyscus are, but also how ubiquitous they are, both in collections and in nature. And that is great for science. Species within this genus vary from acutely desert adapted to total habitat generalists and everything in between. Deer mice are also phylogenetically similar to lab mice, which are physiologically similar to humans. And this makes Peromyscus rodents convenient biomedical models for understanding mechanisms of dehydration tolerance and eventually even developing gene therapies for humans. To start, the McManus lab at the University of New Hampshire looked at the desert-adapted cactus mouse, that is Peromyscus eremicus. This species is endemic to the southwestern deserts of North America in the areas shown here in blue, and may never drink water or pee during its entire life. They're essentially anuric. We generated a high-quality chromosome-level genome assembly for this species by combining 10x, Illumina, and PacBio sequence data with Hi-C data thanks to contributions from DNA Zoo. And we then screened the genome for signatures of selection. We found that functions related to transcriptional regulation and the perception of bitter taste were under selection in the cactus mouse. This suggests that cactus mice may have adapted to low water, high heat conditions through a combination of dietary changes and transcriptional regulation. that is, modulating the relative production of different proteins. We were then curious if this was a common mechanism that many species use to accommodate desert conditions, or if it was unique to cactus mice. So we went back to museum tissue archives and the field and compared the genomes of Eremicus, Crinitis, and Maniculatus, 3 different species within the genus Peromyscus. Both Eremicus and Crinitis are desert specialists endemic to the American Southwest, and their ranges are shown here in red and blue. Maniculatus is a geographically widespread habitat generalist, and its range is shown here in yellow. You can see it's essentially everywhere. That said, the Maniculatus samples used for this investigation were also collected in the Southwest, so it's possible that these individuals are also locally adapted to desert conditions. If we look at the phylogeny here on the right by Gehring et al., we can see that these species are hypothesized to have diverged millions of years ago. But we don't know the ancestral state of these clades. That is, whether the shared ancestor of these 3 species or a subset of these species was arid adapted or not. Therefore, within this framework, we cannot distinguish between convergent and parallel evolution. Parallel evolution is typically observed in recently diverged taxa, where similar selection pressures on similar genetic backgrounds can lead to the evolution or maintenance of similar phenotypes. Whereas convergent evolution has the same outcome, similar phenotypes, but these phenotypes are not typically based on the retention of shared ancestral variation, meaning species may have diverged and evolved in completely different directions before later converging on a similar phenotype that's observed today. Since we cannot distinguish between convergent and parallel evolution here, I will refer to any shared genes or biological pathways as being the result of parallel evolution more generally, but know that it's just a little more nuanced than that. The second important consideration is that the Southwest was not always a desert. Pleistocene vegetation in this region was very different than it is today, and therefore supported a different community of organisms and set of adaptations. Few Peromyscus fossils are identifiable to the species level, so we don't know if these species inhabited these areas historically or colonized after the formation of deserts. However, based on the dated phylogeny, we know that speciation in this group does not coincide with the most recent formation of southwestern deserts, which occurred more than 1 million years ago. With this in mind, we hypothesized that similar genes will be under selection in at least the 2 desert specialists and possibly all 3 species, representing a signature of parallel or convergent evolution. This would also highlight specific genes and pathways that may be universally critical to desert survival. In contrast, limited overlap between the species would suggest there are different molecular strategies to achieving desert adaptation. So we identified significant selective sweeps across the genomes of these 3 species. And if we look at just the nearest protein coding gene upstream and downstream of each sweep, we see no more overlap in the genes identified as under selection between species than we expected by chance. So it looks like there are not common genes that are shared between the desert specialists or between all 3 species. But we did find overlap in the functional groups that were under selection. Enriched GO terms, or gene ontology terms, are essentially, you can think of them as functional groups. And of the many that were identified as under selection, 4 were shared among all 3 species. And this is more than we expected by chance alone. This also hints at the possibility of parallel evolution. Different genes, similar pathways. But if we look at those functional groups in greater detail, they all have to do with ribosomal functionality. So what does this mean? And it could mean a couple things. First, it's possible that ribosomes are super critical for thermoregulation and or dehydration tolerance. And this has been shown in some other mammals. But it's also possible that ribosomes may be involved in other critical biological functions in Peromyscus. We haven't run an association analysis yet with different variables to either eliminate this option or provide more support for the role of ribosomes in these functions. And finally, it could be that ribosomal genes are simply more ubiquitous across the genome. If they're everywhere, they may be more likely to experience selective sweeps just by chance than more rare functional groups. So with this in mind, we've concluded that there's limited to no support for parallel evolution across desert-adapted Peromyscus. Instead, each species may have adapted to deserts by different mechanisms. For example, functional enrichment in Eremicus was restricted to only ribosomes. And you can see this if you look at the labels on the bubbles in the blue plot on the bottom. But Crinitis, which is shown in the red plot on the top, has diverse functional enrichment, spanning osmoregulatory functions, ribosomes, and protein catabolism, all of which are arguably important to desert environments. These results, the lack of evidence for parallel evolution, previous transcriptomic investigations of Eremicus, and the distinct demographic histories of these species together support different adaptive strategies. This is a PSMC plot. It shows the effective population sizes of these species through time, with the present on the left and the distant past on the far right. We're going to ignore the yellow line here for Maniculatus, as the genome sequence used for this analysis is from a captive individual and does not reflect the true demographic history of wild Maniculatus populations, for anyone who is worried. Looking at the other two, the red and the blue lines, we can see that Crinitis in red has a much larger historical effective population size relative to Eremicus in blue. Larger populations are able to sustain a greater volume of genetic diversity or have more standing genetic variation. Therefore, it's hypothesized that Crinitis may have adapted to deserts primarily through changes to protein coding sequences, whereas Eremicus may have relied more on changes to regulatory regions that impact gene expression. Although this is just a hypothesis for now, it's further supported by the close proximity of selective sweeps to protein coding genes in Crinitis, while the opposite is true in Eremicus. Most sweep sites in Eremicus were located thousands of bases away from the nearest protein coding gene. And we look forward to testing this in the future with RNA-seq data from Crinitis. We are now testing the hypothesis that was generated with museum data using live animals. And this is important because it really underscores the role of museums as a birthplace of innovative biomedical hypotheses. Although I don't have a ton of time to go into the nitty-gritty details of this work, I wanted to give you a couple highlights. We are currently using continuous metabolic phenotyping of captive cactus mice to look at changes in energy consumption and water usage under different environmental conditions. Surprise, you now know the answer. We first compared metabolic responses under diurnally variable conditions. So this is what you'd experience in the desert: warm and dry daytime conditions, cool and slightly wetter nighttime conditions. We compared this to cactus mice physiological responses under constant warm and constant cool conditions. These three treatments are shown here in yellow, orange, and blue. The plotted values are the respiratory quotient, or RQ values, across a 24-hour cycle. RQ is the ratio of carbon dioxide produced to oxygen consumed, and is a measure of the types of fuel that an animal is using: carbohydrates or fats or proteins, etc. An RQ value over 1 typically indicates either extreme aerobic exercise or the production of fats from carbohydrates through the process of lipogenesis. We observe RQ values over 1 in cactus mice, but only under diurnally variable environmental conditions, so the yellow plots, and also only during the daytime. Because we know that these animals are nocturnal, we know they're not exercising excessively during this period, so the pattern likely reflects that of lipogenesis. What's really cool about this is that fats produced by lipogenesis can be broken down later in return for a higher volume of water compared to the breakdown of glucose or proteins. Water is both necessary and hard to come by in desert environments, so that makes sense. But the fact that lipogenesis is only observed under regular environmental cycling, warm, hot days, cool, wet nights, suggests that environmental predictability may allow cactus mice to be less careful with their resources. Whereas in the face of uncertainty or extended environmental stress, their bodies favor water preservation, just in case. These new methods of collecting physiology data can also be performed in the field, and we intend to do that next. And if these data are archived with their corresponding voucher specimens or representatives thereof, physiology now represents an additional attribute of the extended specimen. We know how important physiology, human physiology, is to our own health and our own susceptibility to pathogens. So it's the same for other mammals. We can now use these new data streams with the suite of information collected via the extended specimen to shift the paradigm away from the current reactive approach to emerging infectious diseases and public health that cost us $20 trillion+ just last year, and towards a more proactive stance on human health and emerging diseases, of which next generation collections represent a critical component. With that, I'd like to acknowledge the collection of collections that collected, curated, and cared for the materials that made this work possible, including my new employer, the University of Kansas Biodiversity Institute, my old employer, the Museum of Southwestern Biology, the University of New Hampshire, where I am now, and all of my funding sources and phenomenal mentors. And I'm happy to take any questions. Here's a cactus mouse for you.
Thank you, Jocelyn. That was an excellent talk. If anyone has any specific questions, please feel free to put them in the Q&A box. We can address those quickly before moving on. One question I had, as somebody who has a lot of experience with herpetological collections, but none with mammalian collections, is are there any common problems you run into with data acquisition from mammals? For example, like with herps, sometimes the way they're preserved makes it really hard to get DNA.
Yeah, so I think we have similar problems as far as accessing DNA. A lot of historical preservation techniques have fragment DNA and make it difficult to extract. Similar with RNA. RNA, which is how you get transcriptomes, is really, really fragile. And so there's only a few ways like RNA later that we can use to maximally preserve RNA. Otherwise, I think biggest issue is always pests. Pests love to eat mammals. And so it's a constant battle in museums to make sure there are no pests in your collection.
Yeah, that's not as much of a problem, I guess, with something.
And I'm sure imaging for morphology or morphometrics or anything like that also kind of depends on how it was preserved. It's easier to image things that like bones that are dried over skeletal material, but skeletal material, you can do some really neat things now with CT scanning and whole organism.
Yeah, absolutely. Thank you. It looks like we don't have any other quick questions. I see that our other panelist, Brad, has his hand raised, but that might be an accident.
No, it's not. If we've got time, I've got a quick, just natural history question about your mice. That was a good talk. Thank you very much, Jocelyn. So I think I heard you say that one of the mice may never drink water or urinate in its entire life. Could you elaborate on that? I'm a plant person, and that just sounds like, you know,
right.
out of the world to me. So if you just have anything else to say about that, I found that interesting.
Right, it's pretty crazy. We were actually talking about this, that we really need to pause after we say that because everyone has this reaction like, what? But yeah, they don't, because they live in deserts, rain events are fairly rare. If there's water available, they will drink. They have normal physiological function of any other animal. So they can drink, they do have kidneys, they filter water, and they can pee. But if water is not available, they're essentially fine. They lose a large portion of their body weight, about 30% of their body weight, and then they stabilize and they don't lose any more. And so we had hypothesized that lipogenesis was occurring, and we're just starting to get at that as a potential mechanism for endogenous water production. Because otherwise, how are they sustaining a normal behaviors, normal weight otherwise?
Thanks, Jocelyn. It looks like, I'm just trying to see, I think I saw another hand raised from a guest, and if you have a question, just add it into the Q&A for us. That'll be the easiest way to address it. Just want to remind everybody of that. I have a question in the Q&A from Pat. And Pat is asking, what is the age in terms of collection time of the oldest mammalian museum specimen from which you have been able to obtain suitable DNA for your analyses?
For my analyses, I think the oldest samples have been just 1980s. But I know there have been many successful ancient DNA analyses that use fossil data or environmental data to extract from much, much older animals or samples. For example, I know the mammoth genome just came out. So that's a great example of an animal that is no longer with us, but yet we have its genome. So that's pretty cool. And I think the quality of DNA that you need depends a bit on your question also. So you can sequence entire chromosomes from super fresh samples, but you can sequence large portions of the genome. They just might not be a continuous sequence if you have older samples. And as computational infrastructure improves, it makes it easier for us to take those little pieces and kind of fit them together like a puzzle in order to understand the genomes of more ancient animals better.
Awesome. Thank you so much. Okay, we will hear from Kelly Speer now, and I will introduce her quickly. Thank you again, Jocelyn, and we'll have you back in just a little bit for the panel discussion. All right. Kelly, yep, cool, awesome. Okay. Kelly Speer studies the evolution of blood feeding in flies, specifically ways in which symbiotic bacteria enabled their insect hosts to specialize on blood meal. Kelly integrates fieldwork and natural history collections with genomic sequencing, machine learning, and modeling to measure the impact of ongoing and past environmental change on parasites and their microbiomes. She grew up in New Mexico, received her MS from the University of Florida, earned her PhD from the Richard Gilder Graduate School at the American Museum of Natural History, and is currently a postdoctoral fellow at the Smithsonian Conservation Biology Institute and National Museum of Natural History. Please welcome Kelly for her talk on determining drivers of symbiont evolution in a multi-tier hierarchical system.
Thank you very much. Can you see that okay?
Yep, looks awesome.
Okay, great. Thank you so much for having me, and for all the participants for coming. It's almost a year to the day from when I was last allowed to go into the National Museum, and I really appreciate the tenacity of the organizers to host this event, even though the past year has been really difficult. I'm excited to tell you about my research and participate in the panel discussion. Natural history collections are continually reinventing themselves. Collectors contributing to the first natural history collection in Paris in 1635 could not have imagined the way specimens are being used now, just as we cannot predict the innovations that will reinvent natural history collections centuries from now. That we are able to continually derive new information from natural history collections is inherent in their value. For example, advancement of imaging technologies has allowed us to examine previously unseen morphological structures that reflect millions of years of evolution and adaptation. Similarly, the advent of rapid advancement of DNA sequencing has enabled profound changes in our understanding of the relationships between organisms. Through these recent technological advancements, we are realizing that a single museum specimen may actually be much more than a single representative of its species. Each specimen may be a snapshot of a community of interacting organisms, including the host, viruses, bacteria, single-cell eukaryotes like malarial parasites, and multicellular parasites like roundworms. This is a fluorescent in situ hybridization image of bacteria within a fly. So all of the red that you see in this image are bacteria fluorescing within a specialized organ developed by the fly to house these bacteria. And we're able to image these bacteria within museum collections, something that I'm sure the original collectors of those flies couldn't have even imagined. So what is the value of examining interactions between species? We can assess hypotheses that are difficult or impossible to examine otherwise. For example, take the bat fly. So bat flies are obligate host-specific parasites of bats. Here you can see one on its host bat. They are blood-feeding and they vector bat pathogens. So they can vector bacterial, viral, apicomplexan pathogens to their bat hosts. They also have faster generation times than their hosts. And this allows them to act as high-resolution markers for host dispersal, and also allows us to examine hypotheses that would be difficult to assess if we were only looking at the mammal DNA alone. I applied this theory to bats and their parasites in the Bahamas. So these are the islands of the Bahamas, and they're split into two main banks, the Little Bahama Bank and the Great Bahama Bank, which are separated by a deep oceanic channel called the Providence Channel. This channel seems to be an important barrier to bat dispersal, as evidenced by other research in two bat species, Aerophilus ezicorni and Microtus waterhousei. Now, these two species, while they show population structuring across the Providence Channel, they are highly connected between different islands in the Caribbean. So the Providence Channel may be a unique barrier to bat dispersal. In order to assess that dispersal, or those patterns of dispersal more finely, I used bat flies associated with Aerophilus ezicorni to get a higher resolution idea of how dispersal is different across the Providence Channel compared to other areas in the Caribbean. So here's an image of population structuring that we found in bats and their associated bat flies. Here, each bar represents an individual bat or fly, and the colors of the bars indicate the proportion of that individual's genotype that can be clustered into one group or another. So what we see in the bat population genetics is that there is differentiation across the Little Bahama Bank and Great Bahama Bank, in that most individuals in the Little Bahama Bank have a different genotype composition than we see in the Great Bahama Bank. But that structuring becomes more drastic when we look at the bat flies. In the Little Bahama Bank, bat fly genotypes are 90% composed of this light color genotype compared to the Great Bahama Bank, which is more represented by this dark color genotype. To examine this, and just a note, all of these specimens were deposited at the Florida Museum of Natural History and the National Museum of the Bahamas, where the bat skins collected as part of this research are now presented in one of the only natural history exhibits in the whole country, and the only one that includes bats. To examine this difference in population structuring across the Providence Channel more thoroughly, we estimated population size and migration rate across the Providence Channel. So these gray circles are just an image representing population size in the Great Bahama Bank, indicated by GBB, and the Little Bahama Bank, indicated by LBB. So the Great Bahama Bank population size of this bat is larger compared to the Little Bahama Bank population size, and approximately 5 individual bats per generation are migrating across the Providence Channel. So this is consistent with less population structuring that I showed you earlier. In contrast, in the bat fly, we see only one individual migrating per generation across the Providence Channel, and less of a difference in the population size between the Little Bahama Bank and Great Bahama Bank. So what are some of the explanations for this difference that we see in the population structuring between flies and their host bat? Well, bat flies may be reflecting a recent reduction in bat dispersal across the Providence Channel, the changes of which haven't accumulated in the bat genome yet, but have accumulated in the bat fly genome. And this is due to the difference in generation time between bat flies and bats. While 36 bat fly generations occur, only one bat generation occurs. Another explanation may be that bat dispersal is rare, but many bat individuals are dispersed at a time. So because there are long bouts of time in between dispersal events of bats, this allows the bat fly genome to accumulate signatures of population structuring that don't appear in the bat genome. So overall, bat flies allow us to detect dispersal patterns that are difficult to assess if we had only used bat genetics to assess this system. In addition to allowing us to assess hypotheses that are difficult or impossible to examine otherwise, studying interactions between species are necessary for examining other hypotheses. For example, if we wanted to examine arthropod vectored pathogens of mammals, it would be important to understand aspects of the mammal, the arthropod, and the pathogen and how they interact with the environment. Similarly, symbiont evolution requires that we understand something about a microbe and its interaction with its host. If we go back to our bat fly system again, I mentioned to you earlier that bat flies are important vectors of bat pathogens, and their microbiomes can influence their ability to vector those pathogens. We know from model organisms like tsetse flies, which vector human pathogens, that the microbiome of the fly helps the host mount an immune response or even build a protective physical barrier against invading pathogens. In the tsetse fly, this protective physical barrier is actually a lining of the gut, of the fly gut, that is induced by the presence of certain microbiome members, and this makes it more difficult for trypanosome parasites to invade the fly, and therefore makes the fly a less good vector of trypanosomes to other mammals. So changes in the microbiome community in response to the fly physiology, the host physiology, or the environment may have downstream consequences for understanding of disease ecology and transmission in wildlife or in diseases relevant to humans. To examine the effect of environment on the microbiome, I worked with Tiago Teixeira, a Brazilian PhD student, to examine bat flies and bats in the Atlantic forest of Brazil. Here are our sampling sites, and the large section of continuous forest that you see at the top of the screen in green is actually a protected reserve called Regua, and we sampled Regua 3 times. The remaining sampling sites are remnant forest patches separated by agricultural land. And the nice thing about this system is that these forest patches vary in their area and isolation from each other. So we can apply an island biogeography theory as a null hypothesis in this system. If the environment is strongly impacting the microbiome community, then we would expect to see consistent patterns of decreasing bacterial diversity with decreasing area and increasing isolation, according to island biogeography. So here are the bats that we sampled. They feed on fruit, blood, and insects. And here are the bat fly species that we sampled. I'm going to use these silhouettes for the rest of the talk to indicate the 2 different families of bat flies. The silhouette on the left are the Nycterobiidae, and the silhouette on the right represents the Streblidae. And all of these specimens we're currently in the process of depositing in the National Museum in Rio de Janeiro. Okay, so let's look at some microbiome plots. So here, each bar represents a different parasite species, and the different colors represent different bacterial genera and their relative abundance in each parasite species. I've outlined the Nycterobiidae in this black box. And what you'll notice right away is that there's a big difference in the microbiome compositions between the Nycterobiidae and the Strepsilidae. While the streplet flies are dominated by Arsenophilus, which is a primary symbiont, it's hypothesized to provide B vitamins that are missing from the blood meal of flies. We see a relative absence or very low relative abundance of Arsenophilus in the Nycterobiidae. Instead, the Nycterobiid flies are dominated by Wolbachia and Bartonella. Wolbachia can sometimes act as a primary endosymbiont, but it's more often a facultatively associated bacteria or a reproductive parasite of insects that can cause mitochondrial DNA sweeps. Bartonella, on the other hand, is a bacterial pathogen of mammals, and bat flies are the hypothesized primary vector of Bartonella to bats. Other species in the genus Bartonella cause human disease, for example, trench fever, but the species of Bartonella that are found in bats are not, do not infect humans. The other bacteria that are listed here, they play a relatively minor role in the microbiome, it seems, based on their relative abundance, although some of them may be acting facultatively or pathogenically. When we examine how well parasite species predicts the microbiome composition, we find that it's the strongest predictor overall of the composition of the microbiome of bat flies. So how does environment impact the microbiome of bat flies? Well, here are plots of bacterial taxon richness with decreasing area and increasing isolation. And if these bacterial microbiomes were following expectations of island biogeography, we would expect to see a downward trend in both of these plots, but instead we see no trend at all. There's no significant difference between sampling sites in the bacterial taxon richness in these microbiomes. But this is only one aspect of the microbiome composition. In addition to taxon richness, it's also important to examine the relative abundance of bacteria in the microbiome, especially because we found that bat flies have extremely parasite-specific microbiome communities. So while we don't detect any change in their diversity, how does the environment impact their relative abundance? In order to assess this, we used ecological interaction networks. So I'm going to walk you through this example before I show you more networks. Each circle here is called a node, and it represents a bacterial taxon. When two nodes are connected by a line, this means that the relative abundance of those two bacteria were correlated with each other between samples. And we built this network from our samples collected in our first Regua site. So that means when two nodes have a connection between each other, it means their relative abundances were correlated across all of our samples that we collected at Regua. Okay, so here are the other two networks from Regua. You'll notice that the networks all seem very well connected. There's not a lot of examples of isolated neighborhoods of interacting bacteria. Instead, it seems like there's connections to most of the different neighborhoods within the plots. In contrast, when we look at the networks built from smaller fragments, the neighborhoods get much more isolated from each other. We no longer have these bridge nodes connecting multiple neighborhoods within the network. But for those of you who are really eagle-eyed, you might have noticed that our sample sizes vary quite a bit between our sampling sites within Regua and our sampling sites in small fragments. And the n numbers at the bottom left of each plot represent the sample size, the number of parasite individuals that were included to reconstruct these networks. And M is the number of parasite species that were represented in this network. So generally, it seems that the Regua sites support a greater abundance and sometimes diversity of parasites than we see in the smaller fragments, which indicates that these differences in network connectivity are mediated by the parasite community. So that indicates that in this case, habitat fragmentation has cascading effects through the bat, parasite, and microbiome communities. But what happens when we eliminate the effect of network connectivity due to differences in sample size? The way we did this was we decomposed each of our networks into a distribution of motifs. And we took the distance the distances between those distributions and plotted them on a principal coordinates analysis here. So each of these dots on this plot actually represents a network and where it falls in an ordination space relative to other networks that we reconstructed. Each of the dark green circles represent networks reconstructed from samples within Regua, within the reserve. And the light green dots are networks that were reconstructed from samples collected outside of the reserve in the unprotected fragments. And this is a species-informed analysis. So most of these networks are paired with each other, indicated by the lines I've drawn on this plot. So for example, if you look at Aspidoptera falcata, the dark green circle at the very top left corner of this plot, that green dot represents where the network falls in ordination space that was reconstructed from samples of the species Aspidoptera falcata from within Regua. Then if you follow that line to the bottom center of the plot to its light green partner, though that network was reconstructed from the same species, but only using samples collected outside of Regua. So what you'll notice if you now take a broader look at this plot is that generally speaking, all of the dark green dots fall on the left side of this plot, and all of the light green dots fall on the right-hand side of this plot. And whenever we have paired networks from within and outside the reserve, they're always very different from each other. The one exception to this rule is Basilia juquiensis, which is the dark green and light green dots sort of located at the center of the plot. This is the one species we had enough sample size for within the family Nycterobiidae. So the difference in response to habitat fragmentation in this species is likely due to the differences between the microbiomes in these two different families of bat flies. So the overall take-home message from this plot is that even when we control for the effects of parasite community impacting the size of the networks and the connectivity of those networks, we still see a conserved change in the interactions and relative abundance of bacteria in response to habitat fragmentation. Microbiomes may be extremely specific to parasite species, but they still reflect signatures of rapid habitat change. They also, the environmental effect is mediated by parasite species identity. And there seems to be differential susceptibility to pathogens like Bartonella between different families of bat flies, possibly related to the other microbes in those flies. By examining the interactions of the environment, the bat, the parasite, and the microbiome, we gain a better understanding of how this community functions. We expect communities and ecosystems to continue to undergo rapid change in the near future in response to climate change and habitat loss. These changes are associated with an increase in the risk of pathogen transmission and even spillover from wildlife to humans. Natural history collections are designed to link as much possible information to a specimen as possible, including linking information across collections. This makes them uniquely positioned to facilitate studies of multiple tiers of interacting species that are necessary for understanding disease ecology of wildlife. So everything that I've showed you up until this point has relied, in terms of microbiomes, has relied on 16S sequencing. And while 16S sequencing is a really good tool for getting a little bit of information from many members of the bacterial microbiome, it doesn't provide you much information about multiple loci within the microbiome or non-bacterial members of the microbiome. So the next step in this research is applying metagenomes to historical specimens to better understand the interactions between microbes and their hosts. Metagenomes have the advantage of simultaneously giving you genomic information from the host and the microbe. And unlike 16S, it doesn't select only for bacteria. It can reveal information about DNA or RNA if that's what you include in the metagenome or metatranscriptome. The advantages of this additional information is that we can detect coevolution between microbes and a host, or selection of microbial community by the host. We can estimate the functional microbial profile based on the set of genes that are present in the microbiome, which gives us more information about what functions the microbiome is providing to its host, whether they're productive or pathogenic. And we can examine intramicrobiome interactions for disease ecology purposes. So we can finally start to assess these questions of what sort of microbe pathogen competition is occurring within a single host individual. So I've been applying this at the Natural History Museum, the National Museum of Natural History, to blood-feeding flies within the Calyptridae. This includes Hippoboscoidea, so both Nycterobiidae and Strepilidae, the two bat fly families, including their sister taxa, the Hippobosidae, which include ked flies that feed on birds and mammals, and the Glaucididae, which are the tsetse flies that are important vectors of human disease. As a phylogenetically distant transition to blood feeding, I'm also looking at flies within the Musidae. As an initial test to see how much information we can get from a metagenomic approach on historical specimens, we sampled tsetse flies, strepilid flies, Nycterobiid flies, and Hippobosid flies, ranging in age from 30 years since collection to as recently as 2 weeks since collection. And we applied a metagenome sequencing approach and reconstructed the genomes from that shotgun sequencing approach. And the N50 score is a summary statistic of the quality of the genome assembly. So generally you want a higher N50 so that you, in order to have a better quality genome assembly. So what you'll notice is that age doesn't seem to be predicting the quality of the genome assembly that we're receiving from this approach. Instead, it seems that the two species that have the largest genomes, which are the tsetse flies at the top and the Hippobosid flies at the bottom, have pretty poor N50 scores. And this is likely just reflective of not sequencing them deeply enough, but these genomes are also extremely AT rich, and that type of genome is notoriously difficult for sequencers to handle. As an additional examination of how good the quality of this information is, we used a Busco assessment. So Busco has a list of genes that you'd expect to find in your sample. In this case, we use the genes you'd expect to find from flies. So of 3,285 genes that we examined in each of these species, the light blue and the dark blue tell you the proportion that we detected in there, completely detected. The yellow, we have some fragmented information from those genes, and the red is the proportion of those genes that we did not detect at all. So in most cases, we actually did a very good job at detecting most of the genes that we would expect to see in a fly. The one exception, again, is the tsetse fly, Glossina brevipalpus, and that's again likely due to the impact of the much larger genome size and the AT-rich genome. But it seems like from these analyses, we can extend the same protocol that we're using to much older specimens. For example, the ones that are housed in the National Museum, which are, most of them are collected in the 1950s that I'd like to use. So this is a hopeful first step towards collecting more information about the fly hosts and their associated bacteria that we can apply to understand their role as vectors of mammal pathogens. So natural history collections are continually reinventing themselves. We are able to derive new information about complex communities of interacting organisms because museum specimens contain information about pathogens, parasites, and beneficial microbes. By capturing these interactions, museum collections allow us to assess hypotheses we could not otherwise be able to test, especially those related to disease ecology. I'd like to thank these collaborators and funding sources for their help. And I can take any questions.
Great, Kelly. Thank you so much. If anyone has any quick talk specific questions that they'd like to drop into the Q&A box, we can address those now before we move on to the panel discussion. I do have one question from the panel, and that is, yeah, so at the time that you were originally scheduled to give this talk, COVID-19 was kind of just coming onto the horizon. We were just starting to learn about its severity and whatnot. So one pandemic year later, have any perspectives on the utility of museum specimens for tracking wildlife pathogens changed? And is this topic becoming more popular or more urgent in light of the pandemic and what we've all gone through the last year?
Well, I can talk specifically about bat museum specimens, seeing an increase in their use in order to track the prevalence of SARS-like coronaviruses in bat museum specimens, so we can understand their global distribution and identify areas of specific risk. I know there's whole consortia of museums working on this. I believe the Natural History Museum associated with the University of Michigan is actually a partner in those efforts in detecting coronaviruses in bat specimens.
Thank you.
Yeah.
We have a couple more questions in the Q&A. Let me just pull that up really quick. Do you have challenges or face challenges with working with museum samples and specimens from other countries? Or do you have experience with that?
Yeah, I think the most crucial part of working in other countries is involving local collaborators. Inevitably, through their own networking and affiliations, collecting, exporting samples temporarily before you return them to the country are all made easier by having collaborators more familiar with the system and with the field sites. So yeah, every time that I've done international field work, I've definitely benefited from having collaborators locally.
Oh, we have a couple more too. Okay. So, okay. Jeffrey says, he says thank you, first of all, and mentions that the late Vicky Funk and himself from SI Botany Department would regularly discuss approaches to include high school and community college students in primary research. So how would you describe your work at the SI to a high school student interested in science right now?
Well, I think I sort of benefit from a lot of the folklore surrounding bats. You know, people are very curious about bats. So because I sometimes study blood feeding flies that are found on blood feeding bats, I think people are even more interested to find out about the secret lives of vampire flies. So I think relying on, I guess I had to train myself to not be defensive about people's initial responses about bats, and instead invite them into the experiences that I've had in the field using images, or even a lot of times natural history collections make explaining complex research much more easy. Because I find if you can make these esoteric hypotheses a little bit more tangible for high school students, then they're going to be intrinsically motivated to learn more about it.
Yeah, that's great. He says thank you. Laurel asks, have you worked at all with the parasites of the bat flies? Thinking of fungi that can infect the bat flies and potential of bats roosting where the fungus grows to prevent bat flies.
Yeah, Laurel, thanks for that question. No, I have not worked with the fungi of bat flies, but I have found it on some of the specimens that I've collected before. I'm actually collaborating with Danny Hale-Waters, who is an expert on the fungi of insects, including bat flies. So yes, I am not as familiar with the fungi of bat flies, but I have been working a little bit with the bacterial parasites of bat flies, like Wolbachia, that so far we found that it does seem like Wolbachia causes mitochondrial DNA sweeps in bat flies and can make assessing the population genetic structure in these flies a little bit more difficult.
Speaking of Wolbachia, there's a question. I think Wolbachia can skew sex ratio of their hosts. Have you seen evidence of this in the flies? Do you think this would make an impact in the interactions between flies and bats?
That's a wonderful question. I don't know. I haven't looked at it, but I think that would be an awesome next step. Yeah.
Okay, James asks, do bat specimens need to be preserved in a special way to allow viruses to be identified later?
Yes, yeah, Jossie touched on this a little bit earlier, but because a lot of viruses are RNA based, there are some DNA based viruses, but most of them are RNA based. You do need to preserve the bat specimens in a way that will enable preservation of RNA. However, I've been working with some great collaborators here at Smithsonian who are working on developing protocols to get enough RNA out of formalin-fixed bat specimens to assess coronavirus prevalence. So coronavirus is an RNA-based virus. So we can start to get a little creative with our protocols and our sequencing so that we can get the most out of the specimens, however they were preserved.
Awesome. Okay, we'll do one more question here and then we'll move into the panelist discussion. Some of these questions are definitely relevant there as well. But Brooke says, great talk. How many bats have bat flies? Or how much host collecting does it take to get the parasite and microbiome sample size you need for your research?
Yeah, thanks Brooks for the question. So it depends on where you go. Sometimes you will get very clean bats. For example, in the Bahamas, when I was sampling bats there, the Little Bahama Bank had many fewer bat flies than the Great Bahama Bank. And I think that was related to the prevalence of bats in the Great Bahama Bank and also the diversity of bats in the Great Bahama Bank. The Little Bahama Bank has many fewer bat species and also just many fewer bats. So it really depends on where you go, but if you're going to find bat flies on a bat, you're going to find a lot of them. There typically aren't just one or two bat flies on a bat. So I don't have to scrounge so hard.
Awesome. Cool. Thank you, Kelly. Thanks for giving such a great talk and fielding questions. Okay, we will move into our panel discussion portion now. So welcome back, Jocelyn. Jocelyn and Kelly are here. We also, if anyone has questions, feel free to submit them to the Q&A. They can be questions, just general natural history collections questions, or questions about early career scientists, what it's like to be an early career scientist, potential advice that these early career scientists might have, and stuff like that. So any questions that anyone has, feel free to submit them. And I will start fielding those. Okay, one thing that we want to really hit on and talk about in the symposium is how we can improve our data collection efforts when we are collecting specimens in the field. And so something that a lot of people are curious about is how can we better the data that we take from specimens? So what sorts of new data should collections or collectors prioritize? What would help make your field or your research easier, because like you had this piece of information that maybe isn't currently collected with a specimen, but if you had it, then you could maybe expand your research by magnitudes or something. But do either of you have thoughts on that or opinions?
Sure, I'll go first and then pass it to Kelly. I'm sure as scientists, all scientists want more data. There is a balance, of course, between the amount of data you can possibly collect for each collecting event and the amount of work that is. So I think data standardization is probably most important, and then choosing your preservation style to maximize future uses. So in mammal world, there are standardizations for naming and different measurements that we take that are really important, even if you're just looking at whether the mammal has a virus in it. It's taking those measurements, taking important detailed locality information, GPS coordinates of where it came from, when it came from, and its habitat are super important. Yeah, that would be my comment. And then thinking about the future. So not just screening for your one pathogen, but screen for the pathogen and then think, where could this go from here? How much more data can we get from different types of tissues, different types of skeletal material?
Yeah, I totally agree with Jocelyn. In addition, I think the types of broad general collecting that are done at some museums is really crucial for us to capture all of these different interacting species through time. I know it can be tempting to go into the field and capture the things that you need specifically for a certain project, but when we're looking back at how diseases emerge from communities in the past, it's really helpful to understand how different species are interacting in those communities and how it's changed through time. So not only like targeted collection is great, but we also need those broad general collecting that happen repeatedly at a certain site through time in order to understand how those communities have changed.
Awesome. Thank you both. So you both came from the Museum of Southwest Biology at the University of New Mexico, but have, of course, branched out to different institutions. Would you both mind reflecting a bit on your collections training since that point and during that time?
Do you want to go first, Kelly?
Sure, yeah. The Museum of Southwestern Biology is an amazing place to get training, and I feel very lucky to have had the privilege to get training there. Because of the emphasis on broad collecting and collecting as much information from a single specimen as possible. So I got trained on how to collect mammals there, how to collect endo and ectoparasites there, and I got trained on molecular genetics techniques. So the Museum of Southwestern Biology is a master at training students in the extended specimen that Jocelyn was mentioning.
Totally agree. Yeah, the best part about the Museum of Southwestern Biology is their mentality on holistic collecting, which is kind of what I was talking about with the extended specimen, where you're not just sampling mammals, you're sampling entire communities, as Kelly emphasized, as being so important to understanding change through time. And the training, I mean, you do everything from trapping mammals to prepping mammals to installing them in the museum and working in the collections. And then your research as a student there is based on those collections. And as a student, I thought one of the coolest things was contributing through field collecting to future research. So a lot of the research that I did was based on collections that had been made before I had arrived, but I contributed to and built collections that were used by incoming students and new students. So I think that kind of rolling model of you constantly need to be surveying and archiving specimens from all over the place is really awesome. And then seeing how those specimens plug into modern research and all sorts of research. So we had genomics and genetics work going on, but also morphology and stable isotopes. And so it's a really rich community of museum scientists.
Yeah, Jossie, I mean, I'm not sure if this is a similar story for you, but I was not interested in pursuing a science career until I was invited to volunteer and then work at the Museum of Southwestern Biology. And, I had taken science classes and done all that stuff, but it just never really clicked for me that science could be a career and that I would enjoy it so much. And I can remember literally my first day at the museum, walking in, and the Museum of Southwestern Biology is the state repository for Mexican gray wolves if they're killed in the wild. And they were like, hey, we're going to prepare this Mexican gray wolf for curation. Do you want to help? And I had literally never heard anything so amazing in my life. And it totally changed my perspective about biology and science. And so I think natural history collections are an imperative way to get people involved through systemic barriers or just a lack of knowledge about this career. It's a really great way to reach out to them.
I think we had the same first day. If I said, I too prepped a gray wolf on my first day, and it was kind of shocking. I mean, I just had never done anything like that, and it was absolutely the beginning of a new career. And I agree, bringing people in to have those hands-on experiences with collections is super important to building and growing a diverse generation of scientists.
Awesome. Thank you both. All right, so another question. Aside from your publication records and innovations of new technologies, what skills or abilities did you emphasize in your cover letters or your applications that you believe led you to be selected for your current and your forthcoming positions?
Me first. Are we alternating? That's a good question. My current position, I do think my training at the Museum of Southwestern Biology played a large role in that, as well as I know the position that I'm stepping into was specifically looking for a person with a classic museum background that could also leverage modern technologies, specifically genomics. So I think kind of merging these new cutting edge approaches with museums, which are viewed as historical or old, but they're not. So I think they wanted someone that bridged that gap.
Yeah, in terms of information about that I included in application materials or things like that, it is a much different process. I'm not sure if the person who asked this question was more interested in terms of applying to graduate school or getting a postdoc or even a faculty job, which I haven't gotten yet. It's very different based on the stage that you're at in your career, what people are looking for. And so I think the most valuable thing that you can do in learning to write those application materials for different career stages is reaching out to someone who has the job that you want or something similar to what you want and asking them for advice about what information you can include, how you can better build your resume to meet that job. And ask a lot of people, because everyone is just able to give you their own experiences. And the more experience from other people that you can garner, you can make like an amalgam opinion about how best to proceed with those different applications. So I would really recommend finding a mentor and asking them how best to go about different applications, which is what I did. For example, Casey Bell, who's now the curator of mammalogy at the LA County Museum, I met her at the Museum of Southwestern Biology. She was my mentor there, and she's continued to be my mentor throughout my career. So her input has always been very valuable to me in getting my master's position, PhD, and now postdoc.
Awesome. Okay, I've got quite a few here. I'm gonna see if we can stay kind of on the same subject here. Do you have advice for graduate students who maybe haven't worked with collections before, but they're interested in incorporating that into their research? Is there any advice on like the best way to get started with that? I mean, you were just talking about kind of forming those connections, Kelly, with somebody who has experience in where you want to be. And so I could see that as like a very valuable path, but do you have any other thoughts on that? Like how to start putting collections in your research where they may not have previously had a role?
Yeah, I mean, museum collections are there for people to use, right? As long as they're used responsibly. And their museum collections are always looking for people who are willing to contribute specimens to them for the benefit of future researchers. So if you want to work with museums, all it takes is reaching out to one. There, at the University of Michigan, you all have a fantastic natural history collection. So that's an opportunity, but in cases where your university doesn't have a natural history collection, they are distributed globally. So, and they can take specimens from globally. So my advice is to just reach out to a collections manager or curator to discuss ways that you can tailor your sampling so that your samples can be deposited in a natural history collection, or to start discussing ways that you might use their specimens to ask questions relevant to your research projects.
Okay, awesome, thank you. What would you guys suggest for educating people, particularly those who don't have an association with biology, so a non-biologist, about the importance of natural history collections? I know personally that this can sometimes be a contentious point, but how would you say to somebody who has less experience with a collection, like this is why they're important, this is why natural history collections are useful and why we need them and such.
So I'll start. I guess I'll start. I think I know I have this conversation with my parents all the time because they didn't get it. And every time I show them more of my research, they're like, oh, I didn't know museums did that. So I think recognizing that the kind of One Health approach, that everything is connected, humans to animals to their environment. And really the only place that those relationships are fully represented is through natural history collections. And so not only can we study animals, but we can study their associations with their environment, with humans, the entire communities, as Kelly was talking about. And so I think natural history collections are how we get at the complexity of living relationships.
I totally agree with you. And I think it is helpful. People obviously have a negative reaction when they hear that museums have to kill animals. But it's important to follow that up with all of the benefits that come from that. And I think part of having that discussion is also letting people know, like, yeah, we've thought about this. Like nobody takes pleasure in killing an animal. We would only do it if it was absolutely necessary and extremely beneficial to the long-term conservation of that species and for building our collective knowledge about biodiversity. And another point related to that is that not only do we question, make sure that we're applying rigorous standards when assessing how many specimens to collect and things like that, but we've actually done tests to see how broad collecting on a single community through time impacts the mammals there. And that research has indicated that there's no negative effect on the population size or population structure from long-term collecting, which means that if you're collecting responsibly, which natural history museums do, you're not going to have a negative impact on the long-term survival of a species. And I think that helps assuage people's concerns, because it's easy to start to think about collections as, wanting to just gather everything, hoard everything, but that's not what we're doing. We want to take a snapshot of a community, save it for future researchers, and allow people to have access to it. We're not hoarding anything away at all.
I think another really awesome aspect of natural history collections that most of the public isn't aware of is that we're also repositories for other types of samples. So we do go out and actively collect and kind of target things for research, but also like trafficked wildlife that has been like apprehended by the government can end up in natural history museums. Local trappers and hunters. In fact, I've had a couple different African trophy hunters that now they're old, they don't know what to do with things. Instead of those materials going to waste, instead of throwing those away, if they have that date of collection, if they have those permits that show, I was allowed to take this at this time, then we can use that. And so we use a lot of these materials that would otherwise go to waste, and we can use them in science and bring them into collections.
Yeah, and very related to everything that you both just said is, I think that museum collections can be used to inform conservation biology and managers. So if either of you have anything more to add about that, like how are collections actually used to further the conservation of the animals through collecting?
Right, so many ways. I'm sure Kelly has stuff to say also. First and foremost, a lot of collections tell us where species are. That's how we figure out the ranges of animals, and that's also how we figure out that they're changing. So when we're protecting one area, if we resample over time, we may find that an animal's no longer found there. And without that resampling, we wouldn't know that. We'd still be protecting the old area, and it's not doing anything for the animal. So I think resampling through time allows us to update how we approach conservation, as well as obviously there's this whole new conservation genetics angle where we can get at statistics about the level of inbreeding or genetic diversity and how isolated these populations are, like Kelly was talking about in the Bahamas. So there's a lot of differences. I would say the biggest one being where things are and how they're changing.
Yeah, totally agree with Jocelyn. In addition, museum specimens have been used to describe new mammal species and new parasite species that we didn't know existed in the past. And that's because it's difficult to look at something one time in the field and automatically know that it's a new species, especially if it's similar morphologically to a different species. For example, in the Bahamas, Mexican free-tailed bats, which are really common in the United States and throughout North and South America, it looks like actually the Mexican free-tailed bats in the Caribbean are a new species. And the only way that I was able to observe those morphological and genetic differences was through museum specimens by comparing skeletons of Mexican free-tailed bats collected from the mainland in Florida to those found in the Bahamas. I realized that the ones on the mainland were almost twice the size of the ones in the Bahamas, and they were genetically extremely distinct, which there's not really an opportunity in the field where I would be able to hold a Florida bat and a Bahama bat at the same time. So museum specimens provide this really unique opportunity to compare specimens in a much more detailed way.
Awesome. Thank you both. That was great. I'm going to switch gears a little bit because we have quite a few questions about advice from students, it seems. So, all right. Would both of you mind talking a little bit about how you narrowed down your research interests to specific groups of organisms, environments, research questions, etc.? Is this coming from someone interested in applying to PhD programs in the next few years? And they said the prospect of choosing a single project or species to study can be pretty overwhelming, which I relate to as well. So I think a lot of people would appreciate advice on this.
Do you want to go first, Jocelyn, or? Okay. Well, I just want to tell this person that it's totally normal to feel that way, but also keep in mind that what you do for your master's or your PhD or even your postdoc is not what you're going to do for your entire career. It's just what you're going to do for those projects. And once you're essentially just trying to gain skills and knowledge that you can apply to future questions that you want to address. So don't feel like choosing a dissertation project means that you're consigning yourself to an entire lifetime of this one type of research. That's not the case at all. In fact, when I was at the Museum of Southwestern Biology, I only studied small mammals. When I was at the University of Florida, I studied bats and bat flies. And during my PhD, I studied microbiomes. And now during my postdoc, I'm studying not even bat flies and symbiotic bacteria. So it's okay that your research moves around a lot. And in fact, it's really good.
I agree with that. I can't say I intentionally narrowed my research. In fact, I think I intentionally do the opposite. There's way too many things that I'm interested in. I know going into graduate school, I knew I was really interested in mammals. I had a background in animal behavior and wanted to link genetics to behavior because I felt like I couldn't describe behavior. I felt like it was subjective. I was doing ethology and I was like, how can I make this into an analytical bubble? And so I've tried genetics and I didn't think genetics were for me and I loved it. And I think that just experiencing different types of science allows you to find what's most of interest to you. So I always tell students not to focus on a specific organism or study system, but maybe broader questions, because you will learn so much about all of those methods and those systems, and that will tell you more about your interests and the direction that you want to go. But yeah, I don't envy your situation. It's super stressful and there's a lot of options. So talk to lots of people.
I think all of that is wonderful advice. In the same vein, what are maybe some recommendations for someone with a more traditional science research background who's considering transitioning into museums-oriented research, assuming that the person doesn't have a close contact with museums? So have you ever met anyone who had this sort of transition, or did you experience something similar yourselves?
For sure. So reach out to me or Kelly or anyone. We work with museums and would be happy to plug you into the community. It's an awesome community. I definitely have worked with a number of people who are less familiar with museums. When I went from the Museum of Southwestern Biology to New Hampshire, I kind of jumped from natural history into biomedical research. And it was really interesting to me because it was this, I came from a community where you voucher everything and this is how you take data. And I jumped into a community that didn't realize museums would be there to accept those types of samples. And they totally are. So I think just making a connection with a museum or museum personnel can help you plug into those systems, and planning, understanding how you're planning to integrate with museums, whether it is you're collecting materials and giving them to museums or you're using the data, understanding that and communicating that with folks in the museum community can help shape that relationship.
Definitely. And I think a lot of the, I know that I've made some great collaborations through scientific conferences, which a lot of them will be online this year at a reduced price. So it's a good year if you typically don't attend a conference that includes a lot of people from natural history collections. This is a good year to go to those types of meetings, more organismal meetings, and meet those people. And just start a conversation about, how your research could fit into natural history collections. For example, both Jocelyn and I go to the American Society of Mammalogy meeting, which is a really great one for getting to know museum folks. And they frequently have museum-centered symposia, which would be really good to attend.
Yeah, that's a great point with the virtual format everything is in right now. You can kind of get an experience with something that you might not otherwise have the opportunity to see.
The only thing I wanted to add to that question is that I do think museums are kind of a natural part of all scientific work or biology workflows. If you're working with living organisms to any degree, you probably should have a museum as part of your workflow. So thinking about whether the museum is giving you samples or you are giving them samples to make sure that your science is reproducible is super important. And that's across all biological fields.
Somewhat related to that as well, somebody asks, what would you say are the like hot topics or top research questions for museums research right now? What's like hot right now? I mean, I think, sorry. I was going to extended specimen, I think is like a very good example of something. And that's what you're both, working with, so.
Definitely. I think certainly the extended specimen, certainly including more than one different type of data. For example, the way I tackle including more than one type of data is by looking at associated species, right? The host, the parasite, and their microbes. The whole microbiome thing has been exploding in the last 10 years, and our ability to apply that to museum specimens is sort of just emerging. It can be really difficult to control for contamination in microbiome analyses, and our sequencing technologies are getting to the point where we are able to tackle some of those problems that were previously inaccessible. So that's like my hot button topic, I guess, is the microbiome and disease ecology.
I think the elephant in the room is coronavirus. Coronavirus is so hot right now for obvious reasons. And basically, I think all museum collections are looking into their collections and saying, how can we contribute to solving this problem, understanding this problem, figuring out where it came from. And so I know there's a bunch of collections, mammals in particular, that are trying to use the sampling available in museums to help us understand the global pandemic.
Awesome, thank you.
And I also think, like Jocelyn, your talk was the first time that I'd ever heard of physiology being included in the extended specimen. So I was also like, oh, this is new possibilities.
So Jocelyn had mentioned that she just got a new position at Kansas. So congratulations. Would you mind sharing your experience with applying for that position as like an early career researcher? And do you have any advice on when you think you should start applying for jobs of that position? And how do you know you are ready for a higher position? So how do you know you're ready to move beyond postdocing?
I don't know if you ever feel ready. Do you feel ready? Does anyone feel ready? I wouldn't say there's going to be a moment where you suddenly feel ready. I started applying for jobs right away because the job market's really competitive. So the second I started my postdoc in New Hampshire, I put applications in for positions that I really wanted. And I think that's the most important thing, is you will put in a lot of applications and don't bother applying to positions that you don't really want. Apply to positions you want and cater your materials to each position, sending kind of a canned letter or a canned CV to each. They're gonna pass over your materials because it looks like everyone else. So make sure you really pay attention to what skills that position is asking for. And then, I mean, how I got it, I don't know. I just landed the total lottery. I feel incredibly grateful. I had a multi-day interview with a research talk. I did outreach talks and met with the faculty, talked about teaching, all that. I think talking with people and practicing for interviews and practicing your talk is really, really important, because you don't want to come off as a total newbie and you want to feel like you fit in there. But I also think it's important to realize that everyone has a first position sometime, and you may not feel ready, but you're going to make it happen. And so just because senior faculty may be judging you as early, well, you are early, so what? So I think having confidence in the fact that you have a PhD, you're ready, and go for it.
Awesome. Thank you so much for that advice. Okay, I think we've got a couple more here. Okay, I think this should be applicable to both of you, but how receptive, in your opinion, or in your experience, I should say, is the NIH to supporting biomedical research proposals that involve museum collections? So Pat, this question's from Pat. They say it seems like a great approach, but is there any sort of resistance just because it's maybe a different way of looking at answering questions of epidemiology?
Sure, I can start. So the work that I'm doing in New Hampshire is funded by NIH, and it kind of melds the use of museum collections with real field work and new sampling. And I do think you kind of have to be careful with NIH as far as what you're doing with museum specimens, but I think they can absolutely be used and connected to NIH work as long as they are suited to answer the health-based question that you're looking at. And that's an argument that has to be made in the proposal. But I also think even integrating, if you are proposing new collections, saying that your new collections are going to museums and enabling that kind of extended research network can really increase the broader impacts of your proposals.
Yeah, I'm expecting to see a lot of maybe rapid grant calls from NIH focusing on coronaviruses and tracking those using existing specimens. I know there are those grant calls going out through NSF. So potentially those will have like more through NIH, those would be more of require extended information beyond just identifying the distribution of coronaviruses in bats, but understanding I guess building more information about risk factors for whether certain coronaviruses are more prone to pandemic spillover than others.
Great, thank you. Okay, so moving forward now from where you are now, how do the both of you envision your personal research engaging with collections from this point on, but also maybe your future labs, and how do you envision training students in collections research based on what you've taught yourself and what you've learned, but also how might you change your approach as you teach the next generation of collections enthusiasts?
I know. I hope to embody a very similar training experience to what I had at the Museum of Southwestern Biology. I think they really did a lot right as far as training not only mammalogists, but holistic collectors, and holistic collectors that can also use those resources, museum resources, in modern science to answer questions of societal concern. And so I think I do and totally intend to emulate that type of system at KU. My lab is located in the museum, the Biodiversity Institute, and we have a molecular facility there. So I intend to kind of got 3 different lines of research, one being epidemiology based, the other being adaptation in mammals, and then other being kind of a biogeography perspective. And I always say integrative evolutionary biology because NSF loves the word integrative. And it's like, what that really means is I like to do way too much. And so I think it's super fun to combine morphometrics with genomics, with epidemiology. And I think this is where we get some of these complex questions. So I hope to have students that are equally diverse. I hope to have students that want to do some of these different things. And then we can really build bridges between different methods that are applied to museums.
Yeah, I similarly envision myself continuing to do holistic field collecting that I was trained to do at the Museum of Southwestern Biology, both to learn something about parasites and their microbiomes, but also to build information about mammal hosts. Who knows where I will end up. And if I'm not associated with the natural history collection, I definitely plan to build those collaborations with whichever one is as local to me as possible. Because I think natural history collections are key to getting students involved with research who may not know about those opportunities already. And they're also key for doing outreach within the local community and wherever I'm doing field work. It's always very surprising for people to learn about the biodiversity that's in their own backyards. And so it's a great gateway for making people more curious about the environment and nature. So no matter what happens in my future, I definitely envision being associated with a natural history collection.
Wonderful. Thanks. Okay. If our panelists have any final comments or thoughts, feel free to share now. And if anyone else has any final questions, please drop them in the Q&A. But if that's all we've got for now, I can wrap it up for this week.
Well, I would just like to say thank you very much for the wonderful questions, very thoughtful questions, and to the organizers again, and for Jocelyn. It was great to present with you.
Agreed. Thank you so much to the panel, to everyone here, the organizers, and Kelly. It's awesome to see you.
Yeah, and thanks to both of you. This was an excellent panel discussion. It was helpful for me as well as a younger PhD student. So I really appreciated it. And thank you for your talks. They were also very great. Okay, I think that will wrap it up then for this week. Just a reminder, we have 3 more weeks of the ECSS. So we will have 2 more Fridays of early career scientist speakers. And then we will have 1 more keynote speaker in 3 weeks, and that'll be Pam Soltis. So we've got a great lineup. Thank you everyone for attending. Thank you for your questions. And thanks again, Kelly and Jocelyn, for everything.